Hi guys, here we are, chapter 8, nervous system. So this is a pretty big chapter. The nervous system is such an important regulatory system in our body. It regulates almost everything. Endocrine is another big regulatory system. So let's jump in. So let's start with question number 24. Name the two main divisions of the nervous system. So the two main divisions are called the central nervous system and the peripheral nervous system. And I'm going to draw a little, actually, I'll show you a kind of a chart here. Well, let me put this in first. CNS and PNS. CNS is central nervous system, and PNS is peripheral nervous system. All right. So central meaning right in the middle, and then peripheral meaning outside of that. The central nervous system, maybe I should continue, is made up of the brain and spinal cord. So think of right in the middle of the body, okay, centrally located, the brain and spinal cord. And then the peripheral nervous system is all the nervous system outside of the brain and spinal cord. So there's going to be information coming in from our senses, from, external parts of the body, picking up information, but it all has to be transmitted to your central nervous system. And then you have to respond in some way, right? After you process that information, the brain and spinal cord, then information goes out back to structures in which you will respond. All right, the two types of cells. Actually, right here, I'm going to go ahead and bring up an overview. Let's do the overview here. I'm going to bring it up here. Something I drew and then just took a picture of it kind of. So nervous system here. Here's the central nervous system and the peripheral nervous system. Okay. The central nervous system is made up of the brain and spinal cord. Now, what is the peripheral nervous system? Okay, there's all the parts that bring information in to the brain and spinal cord. This is called our sensory division. Okay. So it's our, it's bringing things in from our senses. Right? So info to the central nervous system. Then we have the motor division. So after we process that information, our brain and spinal cord, then we send information out to along motor pathways and to what we call effector organs, to structures in which we will respond, right? So now let's take a look at just the motor division. So just those pathways taking information away from the central nervous system. We have what's called the somatic and the autonomic. Somatic is voluntary, okay? So voluntary pathways, because, we tell our muscles to contract, our skeletal muscles to contract, right? I'm going to pick up this pen. I'm going to speak and say these things, right? So information is traveling along specific pathways that are going to those structures that we can control, okay? Voluntary. And those go to skeletal muscles. Other pathways are pathways leading to structures that we don't have control over, right? Conscious control. These are automatic. So I think of autonomic as automatic, okay? And something automatic is involuntary, okay? We don't actually, think about, I want to do that. Our body does it if it needs to do it, okay? So of those pathways that are autonomic or automatic, involuntary, then we can actually divide that up into two divisions. So maybe back up 1 second. Autonomic, what kind of structures does it go to? It goes to cardiac muscle, our heart. Okay, luckily we don't have to tell it to contract, it just does. And then smooth muscle. Smooth muscle, remember from the last chapter, muscle is found in lining organs, okay, the walls of organs. So we don't have to tell our intestines to squeeze food along, or our urinary bladder to squeeze urine out, or our uterus to squeeze a baby out. Okay, those are all smooth muscles and they're all involuntary. Glands also. So glands, pathways go to glands that tell the glands to secrete their contents. Okay, and that's involuntary. We don't control secreting glands, right? Think sweat glands or something like that. All right, of those, okay, remember we're motor pathways. We're leaving the central nervous system along involuntary pathways, so going to the heart, going to smooth muscle, etc. Now we can actually divide into two different pathways. So one is called parasympathetic and one is sympathetic. Parasympathetic pathways lead to structures under conditions of what we call rest and digest. So kind of going back toward homeostasis, all right? And then the other one is sympathetic pathways. These are going to, again, involuntary, cardiac, smooth muscle, but it's leading or under conditions of what we call fight or flight. So under stressful situations, right? So even we have different pathways, let's say going to the heart, if it's a stressful situation, scary, then certain pathways are going to lead to the heart and tell it to speed up, beat faster, okay? Preparing us to either fight or run away. Other pathways, if the threat is gone, and then we send electrical impulses or information along pathways, the parasympathetic division leading also to the heart. Okay, they can go to the same structure, but this pathway is going to cause it to slow down. All right, we'll take a look at a little more detail, especially this sympathetic, parasympathetic, and talk about, how does the heart know to speed up or slow down? All right, and it all has to do with chemicals that are secreted. All right, so that's an overview. Let's take a look at the different types of cells. Okay, so what are the different types of cells found in the nervous system? And there are two. One type is going to actually conduct electrical messages throughout the body, and the other one is going to provide support. The first one, the neuroglia or glial cells. These are support cells. There's different types of glial cells, and we'll talk about that in question 27. All right, so right now, know that they don't actually transmit messages. The other type are neurons. Neurons are the actual cells of the nervous system that actually transmit information or messages along it. And we'll talk a little bit about how that happens. Like, I'm thinking of what I want to say, then I send electrical impulses or messages to those muscles in my mouth, in my throat, right, or my voice box, that will help me to form words and to say what I want to say. Okay, but I have to tell those muscles to contract in a certain way. Okay, I have to provide air to come out, extra air, which is going to, cause me, or I have to contract some muscles to get air to come out too. And then I form my mouth and my tongue to form the words. Okay, anyway, so, but this is all through the nervous system, okay, which is pretty crazy. All right, so let's take a look at some of these neurons first. Okay, they can't all be the same. We can classify neurons according to two different things. The first thing is according to structure. Okay, so let's talk about neurons classified by structure. Before we actually get into the individual, details of what a neuron looks like. Okay, so in general, we can classify by structure, meaning what does it look like? Unipolar, bipolar, multipolar. Uni means one. Bi means two. Think of like a unicycle, one wheel, bicycle, two wheels. Multi means many. So, I always try to think of a something that you know in your regular language, right? Your words that you already know, because I want to stress not memorizing. Okay, with medicine, and that's why the medical terminology, really take that seriously. It's going to save you a ton of time, and when you have to take board exams and things like that, you're going to have to know these terms and maybe even terms or diseases or structures that you've never heard of before. But if you understood the root words, or being able to compare it to a word that you already know, you could probably figure it out. All right, so multi, like a multi-purpose room. Okay, it's a room that you can do a lot of things in it, right? So many purposes in that room. So again, when you come across any new word, think about, oh, is there something familiar in that I already know? Okay, and that way, again, I want to stress, do not memorize it in the medical field. It's just way too many terms. All right, so let's take a look at what these look like. What does it mean, one, too many? Okay, what am I talking about? I'm talking about projections. So these are neurons, okay? And and really, it's about how many projections come out of this part, the cell body. The cell body is the part that, you think of a cell, it's got a nucleus, it's got endoplasmic reticulum, it's got ribosomes, it's got those things that, make you think of a cell. So a unipolar, it calls it a pseudo unipolar, but there's a single axon that comes out of the cell body, right? Here, a bipolar neuron has two projections coming out of the cell body. Okay, one, two projections. Multipolar has many projections coming out of the cell body. All right, so we can classify it, any neuron according to what it looks like. The other classification of a neuron is based on function. So I'll put here, this is the structural classification. Now I'll do the functional. Oops. Even though it didn't really ask about it, but I'm going to put it here just because we'll be talking about both of these, both classifications. So functional classification. There are also three types of functional classification. All right, so, okay, these are sensory. And I've already talked about this when I did the overview. Brings info to the central nervous system. There is motor. Well, maybe between the sensory and motor, there's something called an interneuron. I should put here. Sensory neuron, interneuron. Now the term inter, I don't know if I've mentioned it. I think I have. Inter means between, right? An international student is a student who is between nations. Intertidal, it's this time between the tides, when the tides are high. I don't know, intersex is between the two sexes. All right, so inter means between. Okay. So interneuron is a neuron that is located between the sensory and motor neurons. Then we have the motor neuron. This one takes info away from central nervous system. Okay, so I'll be using both of those interchangeably. In fact, I'll probably use sensory motor neuron more often. Okay, and so this is what what the structural neuron looks like. So like sensory neurons are mostly unipolar in type. So now we can put it together and really talk about neurons. Let's talk about the different types of support cells, the neuroglia. Wait, we have to go back because part of this question is actually looking and labeling a neuron. So I'm going to actually go ahead and before we get to this, I'm going to draw that. So let's draw a neuron, a basic, what we call a multipolar neuron. And I would, again, encourage you to draw. So when I draw, you draw, okay? And I'll put on here drawings that you can put in your study guide, right? If it's too hard to put in your own drawing, but this is more of an exercise. Again, there's something that happens when you draw. It helps you to remember things more, okay? Because actually when I test students, and say, hey, know this diagram. And if I ask a student, okay, do you know it? And they'll go, yeah, I know it. But then I give them a blank piece of paper and I say, draw it. Unless they've practiced drawing it, they won't be able to draw it. And you may want to try that. Just listen to the video or, watch it and then put it away and see if you can just draw it. Okay, versus just labeling it. Okay, now on the exams, you won't have to draw anything at this point, and maybe someday I'll have students have to draw it, but I don't know. It's a really good exercise. So multipolar. So we're talking about a cell body. That main part that has the nucleus and the organelles and things like that, and then we have our projections that come out like this. Let me just kind of drop it. Okay. All right, something like that. So this is our neuron. Okay. Let me label it. We have these things that stick out, projections called dendrites. Okay, this is the receiving end of the neuron, meaning it's going to pick up the sensory information or whatever information it is and then transmit it along here. This area is called the cell body, as I mentioned. Contains all the organelles and such. This structure is called the axon. The axon is the actual structure that transmits the information over long distances. We can have very long neurons. And then we have the ends, what we call the synaptic knobs, or they've got different names. Oh, I should look in your textbook. Different textbooks call it different things. Let me see. Axon. Let me just look in the book really quickly. I'm going to pause it. All right, so now I looked in your book because it actually is called many different things depending on what it is. So this is called the synaptic knob. You may see it as called a terminal bouton or synaptic bouton or kind of like a button. And actually there's lots of other different terms. And leading to the synaptic knob, kind of the end of the axon, is the axon terminal. Okay. Now I'm going to add something to this. Surrounding the axon of the neuron, we have these specialized cells that actually wrap around the axon. And the cells kind of wrap around and around and around. Think of like insulation on a wire. Okay, but it does have little spaces in between, so I'm going to accentuate that. All right. Oh, let me put it. Now I'm going to label this, and I want you to understand. So an individual cell is one of these glial cells that we'll talk about, but it's specialized. It's called a neurolemmacyte. CYTE means cell. Okay. Another name is a Schwann cell. Now, if this is a peripheral nervous system axon or cell neuron, it would be a neurolemmacyte. If it's in the central nervous system, a multipolar neuron there is called an oligodendrocyte, and we'll talk about that in just a moment. All right, so this is a neurolemmacyte, but if I get all the neurolemmacytes together, it forms a sheath-like structure. We call this the myelin sheath. Okay, so I want you to be able to distinguish here. The neurolemmacyte is the individual cell, whereas the whole structure altogether is called the myelin sheath. Okay, so, kind of same thing, but looking at different parts to it. The spaces here between the neurolemmacytes are called nodes. And this is a neurofibril node, also called a node of Ranvier, named after a person. Same with Schwann cell, those are named after people. That's why it's capitalized. Ranvier, French, I believe. Okay, so I tend to not want to use the Ranvier or Schwann after people because they don't really tell you about what the structure is. They just tell you who discovered it or, labeled it or named it. And so I tend to stick to the term, the actual term. All right, so I hope that's clear. And again, try to draw it and label it, and then you know it. All right, let's go back to our okay. So now let's talk about neuroglia. Oh, I said I'd put some pictures here. Let me give you a couple pictures. Here is the picture from the textbook. Okay. And then here's, what did I call it here? Hold on. My picture. Okay. There you go. So you can use those or not, or, the study guide really is for you and what you put in it, as long as you answer each question. But whether you put diagrams or not, that really is up to you. Okay, I would to study from, but again, it's up to you. Neuroglia. So remember, these are the support cells, and then there's lots of different types, and we're going to talk about all the different types. I've introduced you to some of them. So support cells, there are six types that I'm gonna, wait, let me see what your textbook does. Yeah, so there's six types. And that's what we're gonna look at here. I'm gonna list them. And I'm gonna divide them up into those that are found in the central nervous system and those that are found in the peripheral nervous system. Okay. So we have the neuroglia in the central nervous system, and then we have the neuroglia in the peripheral nervous system. Okay. So the first type is the microglia. Micro means small, and glial are those support cells. And these are actual immune cells. So they have an immune function in our brain. And again, these are central nervous system. The next one is oligodendrocyte. So they form that myelin sheath, but now it's in the central nervous system. So it's an oligodendrocyte. That's the type of cell. They're a little different in the way they look, but their function is very similar. They help information move faster along neurons. So both the myelin sheath in the central nervous system and myelin sheath in the peripheral nervous system. Remember, the myelin sheath is these neurolemmacytes or oligodendrocytes that are forming a sheath along it, but allows the electrical message, to kind of jump from node to node and make it go faster. Okay, so without the myelin, then information moves very slowly. And in fact, there are some diseases and disorders that affect the myelin. So there's something called multiple sclerosis, which causes damage to the myelin here. Causes your own immune system to attack the myelin. And so when that happens, then information passes very slowly along those. And so, it's difficult to walk and to move and things like that. Astrocytes. Astrocytes are associated with blood vessels in the brain. Now, the brain is very protected, well protected from toxins and dangerous stuff in your blood. So there are specialized blood vessels and cells that kind of surround the blood vessels that make it more difficult for material to pass into your brain. And so we call that the blood-brain barrier. And it involves these specialized types of glial cells called astrocytes. I'll show you a picture of these in just a second. Ependymal cells, they line spaces within the brain. So we'll find out that the brain actually is hollow. And it's hollow and it's filled with fluid that actually circulates through the brain and spinal cord and around it. So there's fluid on the inside and the outside of the central nervous system. And what that does is it cushions the brain and spinal cord. All right. Now, there are specialized cells called ependymal cells that are lining these ventricles, these spaces within the brain, and they help to produce and circulate this fluid. And the fluid is called cerebrospinal fluid, or CSF. All right. So those are all within the central nervous system. There's a couple glial cells that are outside of the brain and spinal cord. The first one's called a Schwann cell, or the neurolemmacyte, which we drew. It again forms a myelin sheath, but it does so in the peripheral nervous system. And then the last one is something called satellite cell. Now, satellite cell is specialized cells that are associated with the spinal cord, but in the peripheral part of the spinal cord, outside of the spinal cord. But when we do the spinal cord, I'll show you where they are. So it supports sensory neuron cell bodies. Okay, so the sensory information coming in along those neurons, there are these specialized cells that are found around the cell bodies, and they support that. All right, let me show you some pictures to make it make more sense. Okay. Okay, so let's take a look at some of these. An astrocyte here, remember the astrocyte is associated with blood vessels. And so okay, here we can see them here. They kind of wrap around blood vessels, and they help keep stuff from coming into our brain. Ependymal cells are here, and this is all brain here, and this is a ventricle or a space containing fluid. And so these ependymal cells line these spaces. Microglia is here, and it circulates and helps to, fight disease and fight clean up things and eat stuff. And then here's the oligodendrocytes. The oligodendrocytes wrap around the axons of cells, neurons within the brain and spinal cord. All right. Oh, so this is showing you. This is showing you kind of a neurolemmacyte. And okay, wait. Oh, so here's the neurolemmacytes or Schwann cells. So they use kind of the old term Schwann cell. So don't be confused. All right. So these are the Schwann cells, and then these are little satellite cells found in the cell body of a sensory neuron. So this would be a sensory neuron. Okay, so it kind of surrounds it. All right. So now we're going to get into the physiology. So we just did the anatomy of the neuron and the anatomy of glial cells. Now let's talk about how it works. Up until now, I've just been saying, electrical impulses or electrical messages travel along the neuron and, bring information in or take information out. But what is that information that travels along the neuron? That's what this structure or this question is for. Question number 28. So it asks first for the definition of a resting potential. The resting potential represents the neuron at rest. Okay, so a neuron that's not stimulated. Okay, it's just hanging out there waiting to be stimulated. So that's what the potential is. Potential part of that term means that it is ready to do work. Okay, so resting potential means it's at rest, but it has the potential to do work. So this is it. That's what this is. Okay. And what that means, or the conditions of the neuron are this. So at rest, this is what's happening within the neuron, these conditions. Okay, let me make this a little smaller. You can enlarge this, okay, in yours. Okay, you can take this picture here, this image, remember, go to the ungraded resources and go to the lecture images or the textbook images. Can't remember how I put it, but lecture images. And then I have this for you, and you can cut and paste and put it in your study guide. But you can alter the size, right? If you want to make it bigger, if you want to make it smaller, you can do that. But I'm going to minimize it here so it doesn't take too long. All right. So here, what is happening in the neuron? And I'm going to draw a picture here depicting these conditions that I just wrote out. Okay. All right, so let me read it first. When we look at a membrane at rest, meaning it's not stimulated, the membrane is what we call polarized. That means it's not the same on either side of the membrane. Okay, so we're looking at the axon. So think of it as a cell. So there's going to be a membrane, and there's going to be an inside and an outside part of the membrane. Okay, so maybe, all right, well, let's just look at this. All right, so think of when something's polarized. What does that mean? If you and your children or your parents' views are polarized, meaning they're like opposite, they're different, right? On a particular topic, there's polarization. You believe something, but your parents or your children believe something else. Okay, you're polarized. As you move together, let's say as you talk about the issue and you come to more of an understanding, right? Your opinion might change and their opinion might change, and you might kind of come together. We depolarize the situation, meaning we're less polarized. So we're coming together and we're more, coming toward each other and making it more equal, our opinion. Well, this term is also used in the neuron, okay? So what is that polarization? Sodium, there's more sodium ions outside of the cell, less on the inside. And then there's more potassium ions on the inside of the cell versus the outside. So that it's polarized according to those ions. Okay, so more sodium on the outside, more potassium on the inside. It's different on either side of that membrane. And so what that looks like is this. There's more sodium out here. There's more potassium inside. Now realize that there's sodium inside and there's potassium outside, but this is just an overview as far as comparing these two particular ions. The A negative are just negatively charged ions because, for the positively charged potassiums, there's negatively charged ions that kind of balance it out. And also here, should have put them outside too. There's also negatively charged ions here that counteract that. So we don't have a lot of positive charges on the inside or outside. All right. So, but it's balanced as far as its charge, but just the type of ion is different on either side. Now, this little thing, because there's people who actually have measured the inside, positivity or negativity on the inside as compared to the outside. All right. So what they found is when they measured on the inside, it's actually more negative on the inside than it is on the outside. That's what this means. And so they found it's about negative 70 millivolts on the inside. And what they found is that it was due to there's potassium channels, the channels in the membrane, were a little leaky. These channels were leaky so that the potassiums would just kind of leak out and it would move along its concentration gradient, right? So because of diffusion, where there's more, okay, the flow of anything is going to flow to where there's less. So if these little channels are kind of leaky, then the potassiums are going to automatically start leaking out, okay, going from a high to a low concentration. Now, why don't the sodiums leak? Well, because the sodium channels aren't leaky. These are just potassium channels are a little leaky, so the sodium leaks out. And as the sodium leaks out, notice the positive charges are leaving, which leaves more negative charges on the inside. Okay, so that's why at rest, it's more negative on the inside of a neuron. Okay, most cells, our muscles too, conduct electrical impulses. All right, and they calculate about negative 70 millivolts. Okay, so that is at rest. Let's talk about what happens when it's not at rest, meaning when it's stimulated. So let's stimulate our neuron. Okay, I'm going to do B. I'm going to call it B. And we're going to take a look at what happens when it's stimulated. So I'm going to call this the action potential. The action potential is a stimulated neuron where stimulated neuron, well, I'll say that I'm going to draw what it does. That can, that in which information is transmitted along the axon. All right. So this one I'm going to draw, and you may need to draw this because, well, I do have a picture actually. What does it look like when it becomes stimulated? All right. When it's stimulated, actually sodium channels open up because over here at rest, the sodium channels are closed. But when stimulated, the sodium channels open and the sodium comes rushing in to the inside. So what does that look like here for it to measure the inside? that means it would go way positive because all the positively charged sodiums are coming into the neuron. All right. And that is called depolarization. So sodium channels open, sodium rushes in, we get a big flux of positive ions coming in, and that's called depolarization. Okay. Now, eventually the sodium channels close, and then the potassium channels are going to open. So once the stimulation is over, the potassium channels open. All right. And the sodium channels close, and then the potassiums leave the neuron. Okay. And so as positive ions leave, then the inside is less positive, and we call this repolarization. And this is trying to go toward resetting because a neuron can't be stimulated again until it's reset. But the thing you may or may not have noticed is, wait, if sodium's coming in and potassium's leaving, that means it's like opposite of a neuron at rest. Okay, now the sodiums and potassiums are in the wrong place. And so that neuron won't be able to be stimulated until we put it back to where it belongs. And one of the ways we do it is we have a pump. There is a sodium potassium pump in the membrane that's going to pump the sodiums out and the potassium in, and it's done that's not quite equal. Okay, 3 sodiums for every potassiums are pumped. Now, almost every cell in the body has sodium potassium pumps because we always have to get our ions in the right place. And once it's pumped into the right place, then we can re-stimulate it. Okay. And so this wave of depolarization here as the sodium channels open and repolarization as the sodium channels close and potassium channels open. Okay, and it causes this like wave of opening and closing that happens along the neuron. Okay, and that is what moves along the neuron. The opening and closing of these channels, we call that an action potential. Okay, it's this wave of depolarization and repolarization of the membrane. So it's pretty complicated as far as how information moves along neurons. And you think about it, when you think about it, we totally take this for granted. So, to contract a muscle, to talk, move, to do anything really that involves the nervous system, it requires this information or this action potential moving along the neuron. Okay. And so that is the physiology of a neuron. Okay. And there's actually more, but, trying to simplify it a little bit. Okay. The next one, now we're going to move into the central nervous system specifically. We're starting with the spinal cord. Okay. Let's take a look at the structure of a spinal cord and label a spinal cord. We're going to look at a cross section. Now, a cross section is if we take our spinal cord, which is about, I'm going to turn off my paper, which is about the width of your pinky finger. Okay, so your pinky finger here, I've kind of got fat fingers, but your pinky finger, if we chopped it and then looked down into it, that's a cross section or a transverse section, right? And so what does the spinal cord look like if we do that? And that's what I'm going to draw right now. It's a transverse section or cross section of the spinal cord. Okay, so here we go. And remember, we're talking about the width of your pinky finger. Okay, so something like that. Now, the inside is a little darker in color than the outside, and it's got this kind of butterfly shape pattern to it in a cross section of a spinal cord. Okay, and that would be what your spinal cord looks like. Just like that. Now, let's label a few things. There's a hole or canal right down the middle called the central canal. The central canal, and in it circulates CSF, cerebral spinal fluid, circulates within that. This all is gray matter. Let me look at our list. What do we have to know? Central canal, gray matter, white matter. So all this is gray matter here. Because it's a darker color, and over here is white matter. Now, gray matter is actually parts of the neurons. It's the cell bodies part of it, and the dendrites and cell body part of neurons. And then the white part are the myelinated axon. Okay. So there are different what we call horns in here. This is the posterior. Okay, you don't need to know that. So this is the posterior side of the spinal cord. This is the anterior side. Okay, in other words, if the spinal cord's coming out of the paper straight at you, okay, then this is the front of the body and this is the back of the body. All right. How about now, how do we communicate with the spinal cord? There has to be pathways to bring information in. In other words, neurons can enter and exit the spinal cord. And it does so through structures like this. All right, so this is how we communicate with the rest of the body. This back here is called the posterior or dorsal root. So this is along the back, right? So this is the root leading into the spinal cord. This is the anterior or ventral root along the front, which takes information out of the spinal cord. And here we have where they come together, it's called the spinal nerve. Now the spinal nerve is bringing, information in and out to and away from the spinal cord and going to other parts of the body. Okay, so again, along the spinal cord, we have spinal nerves that leave the spinal cord. So if I were to draw, here's our brain, here's the spinal cord. Okay, the spinal nerves are found along the spinal cord like this. And these are spinal nerves. And these communicate with the rest of our body, sorry. Okay, there are 31 pairs of spinal nerves leading to and away from the spinal cord. I'm going to finish this. Let me finish this drawing because I want to put in the neurons and how they communicate with the spinal cord. So if you touch a hot stove, sensory information comes into the spinal cord through the posterior root. It's a sensory neuron. Okay, so sensory information comes in through the backside, okay, through the dorsal or posterior root. And this is a sensory neuron. So it's bringing information in. Now, in the middle there, we may have what's called an interneuron. Okay. Remember, it's a neuron between the sensory and the motor neuron. And it's completely within the spinal cord or completely within the central nervous system. The last neuron is the motor neuron. Okay, because we touch a hot stove, we have to respond. So this is our motor. And how do we respond? We contract our muscles in our hand and in our forearm, and we pull our hand away. So we touch a hot stove, that pain information comes in and then goes out the motor neuron through the anterior root and then goes to our muscles, right? Our skeletal muscles, and we contract our muscles and we lift our hand away. How fast does that occur? It happens so fast, right? You touch a stove, whoa. But information has to go all the way to your spinal cord and back out to your muscles, and then you contract your muscles. We talked about muscle contraction, the 10 steps of muscle contraction. All that has to happen before you even pick up your hand. All right. So the human body is amazing. It's amazing, right? To be able to do that without even thinking in a split second. So just to at least gain an appreciation for what your body can do. All right, so that's the spinal cord and, the different parts of the spinal cord. And then, giving you an idea as the sensory and motor neurons are coming and going. These are the spinal nerves that spinal cord, go to the spinal cord. And so let's say, let's talk really briefly about spinal cord injury. So let's say you have a spinal cord injury, like here. Okay, so remember, I didn't draw them all. There's, a bunch of spinal nerves, 31 pairs, right? And they're all going to different parts. These can go to the arm. These can go to, let's say, the diaphragm. The diaphragm is the main breathing muscle. And then down here, we have more. Oh, let's make our spinal cord longer. All right, then down here, some go to the legs, right? So this isn't very accurate. There's actually a space here. But anyway, just giving you an idea of how this works. Let's say you have a spinal cord injury here. What happens? This means we don't get sensory information going to the brain. Okay, so we don't have any sensation. And then we don't get motor information coming from the brain to the arm and to the legs. So this cuts off information for everything down below. And so, but you're alive, you get to breathe. Okay. What if the spinal cord injury is really high and it cuts off information to the diaphragm, your main breathing muscle? So sensory information doesn't come in, motor doesn't go out. Usually people die very quickly. There's a saying that says, C4, live no more. Because people with a high spinal cord break above the level of C4, okay, cuts off information to the diaphragm. And in fact, there's a special nerve called the phrenic nerve that comes off of this. Phrenic nerve to the diaphragm is disrupted. Okay, and so stop breathing. People stop breathing. So unless someone's ventilating for them, they typically die quickly, within minutes, right? All right, so that's why this information is important for you guys. When you're looking at records, you're looking at, how do you transmit this information into records, right? To have an understanding of the implications of what kind of injuries and what it means to have if they talk about different spinal nerves, okay, that are involved in this injury, then you know what kind of paralysis, or sensory loss or motor loss that will be happening, right? So the further down you go, the more spinal nerves can communicate with the brain, all right? So it is very important. Okay, so let's go back. All right, and then I'm going to put this little picture here. I hope you drew it. Okay, well, there's my little picture. And then here's some more information about the spinal cord, how many pairs. And then something that I didn't really talk about, which is the plexus. So let me put that here. There's what we call cervical plexus, brachial plexus, lumbosacral plexus. And so this is kind of what I was drawing. If we look at our picture here. So the cervical nerves here will lead to a plexus, okay? The upper cervical nerves, C1 to C4, C5, form what's called the cervical plexus, which then goes to the diaphragm. The lower cervical plexus, and actually one of the thoracic or nerves, some of the cervical nerves and thoracic nerves, form what's called the brachial plexus, and this goes to the arm. And a plexus, and maybe I should go back. A plexus is a kind of a jumble of spinal nerves. So some of these spinal nerves, like the cervical spinal nerves, come off and they'll form a network here, what we call a plexus. So this is the cervical plexus formed by the first cervical vertebrae, or the first cervical spinal nerve. All right, it forms a plexus, and then we have nerves that come off of it, like that phrenic nerve. Okay, then the next ones form the brachial plexus, a different jumble. All right, and then those will go to different parts of the arm. Okay, so this goes to the arm here. And so that's what the plexus is. It's a group of these spinal nerves that form a kind of a jumble, a little network. And that's important because, if like this one cervical nerve, the cervical nerve number one, or spinal nerve, goes to, one structure, if just that one went to the diaphragm and that got damaged, that would be it. You would die. But with the plexus, if there's damage to one of these spinal nerves, then information can still travel along other pathways. All right. So just those networks make it easier or, better to be able to communicate and with different areas easier. All right. Let's go back. Okay. So, oh, whoops, this was that next question. How many plexuses? And that kind of goes with this. Oops. Okay, 31 pairs of spinal nerves. And I tell you how many of each. There are 8 cervical spinal nerves, 12 pairs of thoracic, 8 pairs cervical, 12 pairs thoracic, 5 pairs of lumbar, 5 pairs of sacral, 1 pair of coccygeal. So that should be 31 pairs. And then the different plexuses are the cervical, brachial, lumbosacral plexus. Okay. All right. The next one. Let me put that. I'll leave that here. Yeah, that's a good place for it. Maybe, actually, let me put this question above that or before that, because there's space here. Okay, and then this picture goes with that. Let's take a look. What is a reflex? You've heard of the term reflex? And a reflex arc. So a reflex is an automatic, unconscious response to a stimulus. I talked about touching a hot stove. That's kind of a reflex. If someone hits your knee with a little hammer below your knee, you kick out your leg. That's called the patellar reflex. There's different reflexes in our body that we can utilize if we're testing for nerve damage or something like that. Neurologists would use the knowledge about different reflexes. So the reflex is something automatic that you do without thinking. And there are different parts to the reflex arc. There's a receptor. I drew a really little picture here. Bigger. All right, we have a receptor. So we have to pick up the stimulus. So the stimulus, let's say, what did I do here? A little bug. Okay. A bug crawling on your skin. That's the stimulus. You have receptors in the skin that pick up that little tickle tickle. All right. And that's sent along sensory neurons into the spinal cord, which connect with maybe an interneuron and then a motor neuron, which goes out to muscles and you flick your hand. Okay. All right. That's a reflex. So we have a sensory receptor responding to a stimulus. I should put stimulus. Stimulus to the sensory receptor. From the sensory receptor, we send information along sensory neurons into the spinal cord, and then that connects to an interneuron in the central nervous system, which connects to a motor neuron, which then takes information to what we call the effector, which could be muscles or glands. Okay, we can flick our hand or we can secrete something and then we respond. We have some kind of response. All right. Okay, there we go. That's a reflex. Let's talk about the brain. Okay, so the brain, I always feel a little like it's hard to talk about the brain because there's so much to the brain. Maybe I'll post a video, looking at a brain, either a model or a real brain, and looking for some of these things. Okay, so let's take a look at a brain. I'm not very good at drawing the brain. That's one thing that okay. So, maybe we'll actually look at the outside of a brain first. Take a look at this. So there's different parts to the brain. There are 4 main parts to the brain: the cerebrum, the cerebellum, the diencephalon, and the brainstem. Okay, again, cerebrum, cerebellum, diencephalon, brainstem. Those are the 4 main parts of the brain. So this part that we're looking at here is the cerebrum. So here's the cerebrum. What we think of really the brain is actually the cerebrum. And the cerebrum is divided up into different lobes, okay, based on the bones that are overlying it. We have the frontal lobe of the cerebrum. We have the parietal lobe. We have the occipital lobe and the temporal lobe. And there's different functions that are concentrated in these different areas. But the thing about the brain is that it undergoes something called, well, it has this characteristic of something called plasticity. I don't know if you've ever heard of plasticity or something being plastic. It means that it has the ability to be flexible. So damage to one area, sometimes other parts of the brain can take over function for those damaged areas if it's not too bad or depending on what it is. So, things like planning and intelligence is found in the frontal lobe. The temporal lobe is like hearing and smell. Parietal lobe, we get a lot of sensory information in, especially along this little ridge here. And then the occipital lobe is vision, concentrated there. All right. We do have like speech, which is kind of over here in this area, which is part in the parietal, part in the temporal lobes and things like that. All right. Then we have the cerebellum down here, which is an inferior structure to the cerebrum. All right. Let's take a look at the other picture. All right. Then when we take a look at a sagittal view, a mid-sagittal view, when we look at the cerebrum, let's do the four main parts. So we have the cerebrum, the cerebellum. The diencephalon is right in the middle here. So right here is the diencephalon, right here in the middle. And then we have the brain stem down here. So kind of anterior or in front of the cerebellum is the brain stem. Okay, now let's look at the different parts within these four main parts. Okay, so it says, what are the four main parts? And then I have in parentheses some of the structures within those main parts. So let's start with the cerebrum. We have the longitudinal fissure. So the longitudinal fissure is a crack that divides the right and left hemispheres of the cerebrum up. Okay, now it's not complete, meaning we don't completely separate the right and left cerebral hemispheres, we call them, but there is this crack there that kind of separates them a little bit, right? We did the four lobes and now the corpus callosum. So let's look at this picture. There's this band of what we call white matter. This is the corpus callosum. The corpus callosum allows communication between the two sides of the brain, the right and left hemispheres. All right. And so information passes back and forth across that corpus callosum. All right. Next, we have the diencephalon. The diencephalon's right here in the middle. And if we take a closer look, all right, we have the part right in the middle here is called the thalamus. The thalamus is a relay station. So as information comes up, all right, you know that bug is walking on your skin, the information comes to the spinal cord, but eventually you say, oh gosh, that bug is crawling on me, right? There's realization of what that is. Yeah, there's a reflex that makes you wave your hand, but there's got to be information that comes up into the brain, and the thalamus is the relay station that sends it to where it needs to go. And so that's right here in the middle. Below it, okay, hypo means less than. So hypothalamus is this little area here right below it. Now, this is an important endocrine structure. It doesn't look like much here on the brain, but it is an endocrine structure. All right, and then hanging off that is the pituitary gland. Again, an endocrine structure. All right, so that's the diencephalon. Here's the cerebellum. Here, there's this pretty branching white matter on the inside of the cerebellum called the arbor vitae or vitae. It means tree of life. Arbor is tree, and vitae is life. So tree of life, because it's just this pretty branching pattern. And then here is the brainstem. The brainstem is made up of the midbrain, the pons, and the medulla oblongata. Okay, the midbrain, the pons, and the medulla oblongata. And those are the main parts of the brain. Okay, let's take a look at the next question. Let me make this smaller just so we can take up less space. Again, you can make it as big as you want. Okay, now how do we protect the brain and spinal cord? We do that through coverings. There are coverings that are found around the brain and spinal cord. These are called the meninges. Okay, meninges are these coverings, and there are three of them: the dura mater, the arachnoid mater, and the pia mater. Dura means durable, and mater means mother, so that translates into tough mother. Okay, so when we look at our layers, it's like leather. It actually is like leather. So here's the skull here, and then we have the dura mater, the tough mother. Deep to that is the arachnoid mater. Arachnoid means spider, right? Arachnophobia is a fear of spiders. So arachnoid mater is a layer that looks spider webby, okay? And then deepest layer is the pia mater, which kind of actually adheres right on the surface of the brain. Well, you know, we don't really peel it off or anything, all right? So that's the pia. Pia means delicate. And so not only does it surround the brain like this, but it's continuous with the spinal cord. So it goes around the brain and then around the spinal cord. So is that important? Meninges, meningitis. So you may see the diagnoses of meningitis and have to deal with that. Meningitis is infection of the meninges, okay, which is serious business. Right, so here are the layers again. All right. Path of CSF. So I talked about cerebrospinal fluid, and it's that fluid that's found and produced within the brain, and then it actually circulates through the brain and around the brain and spinal cord. So there is a path that it takes. This. Okay, we're going to take a little closer look. Now, inside the brain, as I've mentioned, it's hollow, right? So there's spaces within the brain. Make it bigger. All right. So inside here are these spaces. These are ventricles. And if we were to follow the path of CSF, it goes in different places. Okay. So 1, 2, these are the ventricles. This is called the lateral ventricle. This is the third ventricle, but don't really worry about that specific parts. But these are spaces in the brain. Then the cerebrospinal fluid flows down this canal in the midbrain. This canal is called the cerebral aqueduct. From the cerebral aqueduct, it will go into another space called another ventricle. It's the fourth ventricle. And then from here, it'll make its way out through little openings called apertures. Okay, and then also circulate around, down around the spinal cord. And all right. And then, so the yellow is the cerebrospinal fluid. The blue is our veins, blood. Okay, because the goal is to get the CSF back into the blood. Okay, so actually CSF, cerebrospinal fluid, is generated from blood because there's blood vessels here, and fluid leaves the blood and forms the cerebrospinal fluid, which then circulates. Now, part of the cerebrospinal fluid goes down the central canal of the spinal cord. Remember that little hole when we do a cross section? Well, it's a canal, and so the CSF goes down that canal, and then it leaves and goes around the spinal cord as well as around the brain. Okay, so it's going to circulate around the brain. Now, what space is that? Between what meninges is it? It's between the pia mater and arachnoid mater. All right, so Actually, let me draw that. It's a little easier to. All right. So if we look at the meninges, we have the outer dura mater. Okay, the outer dura mater, it's tough like leather. We have the inner arachnoid mater, and then we have the deep pia mater. Now the pia mater actually extends down into the little what we call sulcus of the brain, all the little cracks, grooves in the brain. Okay, so here's our brain. And then the pia mater sits right on top of that. And it actually goes down into the cracks. All right, so if we have the dura, the tough mother, we have the arachnoid. Oh, arachnoid mater. We have the pia mater. But between the arachnoid and the pia mater, we have this space. Okay, or I'm going to put it right here. This is called the subarachnoid space. And it's here where we have the cerebral spinal fluid. Okay, so it circulates here. Now there are areas where the arachnoid mater pokes up through the dura mater. Okay, in the top of your head. So now we're going to look to see where does the arachnoid and where does the CSF circulate. It's going to go this way. And this little thing that pops out is called the arachnoid villus. Okay. And so the CSF is going to come out through here and it's going to go back to the bloodstream in a vein and dump back into a vein. Okay. Does that make sense? All right. So it circulates in the subarachnoid space through these little structures called villi and then back into the veins that are in your head. So if you have a subdural hematoma, a hematoma is a collection of blood. Let's say you hit your head really hard and you have a bleed in the head, but it's a subdural hematoma. That tells you where it is. Hematoma is a collection of blood, but where is it? Subdural. It's under the dura. It's between the dura mater and the arachnoid. Okay. If you get a what else? Let's see. Epidural. Okay, you get an epidural. Where do they put the anesthesia? Epi means above and dura. So it's above the dura mater. Okay, so these are terms that are going to appear in medicine. So understanding what it is you're looking at, what it is you're reading, is very important. All right, so let's go back. Hope that was helpful. Go back to our pictures. Okay, so now hopefully this makes sense. Now we're here's a little arachnoid villi up here. They call it arachnoid granulation. The granulations are a whole bunch of the villi. An individual one's a villus. So this should actually say the arachnoid villus. All right, but a whole bunch of them, and they're all feeding the CSF back into the vein here in the top of the head. All right, let's finish up. How many cranial nerves are there? Okay, how many cranial nerves are there? There are 12 pairs of cranial nerves. 12 pairs of cranial nerves. Let me get a picture. Yep. Yeah. So just as Spinal nerves leave the spinal cord. Cranial nerves leave the brain. There are 12 pairs. All right. Now, do you have to know them? You should know them. Let me go through them really quickly. The first one is the olfactory nerves here, optic nerves, oculomotor nerves right here, trochlear nerve, this little guy right there, trigeminal, tri means three, so it's going to branch into three different places. We have the abducens right here. Oh, sorry, wait. Abducens is right here. It's small, my eyes aren't that good. All right, abduce, maybe I'll make it bigger. Okay. All right, abducens here. Then we go to, let's see, vestibulocochlear. Oh, wait, we skipped the facial. Oh, then facial nerve here. Facial first, then vestibulocochlear, glossopharyngeal, then the vagus nerve, the accessory nerve, and then we end with the accessory nerve here and the hypoglossal down here. Okay, so there's a mnemonic. Maybe I'll put the mnemonic here to help you remember. Oh, once one takes the anatomy final, very good vacations, are heavenly. All right, so olfactory is smell, optic is vision, oculomotor helps to move the eyes. Trochlear also helps you move the eyes. Trigeminal goes to the face and other structures, three places. And abducens also moves the eyes. Facial goes to the face. Vestibulocochlear goes to the ear, the vestibule and the cochlea, which are important for equilibrium and hearing. Glossopharyngeal goes to the throat and the tongue. Vagus nerve goes to lots of different places internally. Accessory nerves go to the neck and throat. And hypoglossal also goes to the tongue and the neck area. All right, so those are the cranial nerves. I do want you to know them the way, let's see, know the mnemonic so you know the order, and they're numbered 1 through 12. So the olfactory nerve is cranial nerve number 1. So that's what the little Roman numerals are here. And then, optic is number 2, cranial nerve number 2, oculomotor is 3. I'm not quite sure how I'm going to test you on that. I'll let you know when we get to that exam. Okay? All right, but I do want you to know the 12 pairs of cranial nerves. All right. Autonomic nervous system. I talked about at the beginning, going over the autonomic nervous system. And just to review, it's part of the motor division, right? We're in the peripheral nervous system, the motor division, so just information leaving the spinal cord, brain and spinal cord. And then it divides into, well, there's 2 different types of pathways in the autonomic nervous system. So remember, these are involuntary pathways leading away from the central nervous system and used or utilized in different situations. So and these 2 different pathways are the sympathetic and the parasympathetic divisions. One thing I put here, it's a 2-neuron system. So let me draw this really quickly. Okay, so when I see a 2-neuron system, so this is the autonomic nervous system. Sometimes we call it the ANS. So remember, these are involuntary. So it involves 2 neurons. So here's neuron number 1, and here's neuron number 2. And we're going to a destination. So this is the central nervous system here. And this is some destination. So maybe it's the heart. Okay, it has to be an involuntary structure. All right, so this is the autonomic nervous system. It takes 2 neurons, it's going to a structure, and it's involuntary. Let me define a couple things. So this place where the 2 neurons meet, this is called the ganglion. I think of it as going on a trip. You're getting on an airplane, and that airplane, okay, remember it's a neuron. So that airplane, you have to have a layover. Okay, you didn't get a direct flight. So your first flight takes you to another airport, which is the ganglion. You get on a different airplane and you go to your destination. But at the ganglion, there's other airplanes, there's other cell bodies of neurons that are leaving and going other places. All right, but you're just connecting to this one. So think of it again as a connecting airport, the ganglion. So what would you call this flight? This is the pre-ganglionic flight. And this one would be the post-ganglionic flight. Right? Now, between how we communicate between neurons and between the neuron and a structure, we saw it in muscle contraction, right? It's neurotransmitters. So we have neurotransmitters here that are released at the, at the junction here between what we call it a synapse. And it goes from an electrical thing to a chemical thing. And then these chemicals bind to the next neuron and cause electrical impulse. When we get to the end here, we secrete more neurotransmitters, which travel across and bind to the whatever it is our final destination is, in this case, the heart, and it's going to bind and cause an action. It's going to cause the heart to speed up or to slow down. Okay, so now let's compare the sympathetic and the parasympathetic division. The sympathetic is fight or flight, stressful situation. We're either going to fight or we're going to run. And I'm going to draw it. This is the anatomy of the sympathetic nervous system. Our first flight or our first neuron is very short. And let's say we're going to the heart. So if it's fight or flight, we want to speed up our heart. The second neuron is very long. You see how that works? Okay. So the first neurotransmitter is acetylcholine. That's a neurotransmitter. But the second neurotransmitter is different. This is called norepinephrine. Also called adrenaline, or, similar to adrenaline. All right, and it's the norepinephrine that's going to cause the heart to speed up. This is the preganglionic axon. This is the postganglionic axon. Here's the ganglion. All right. What does the parasympathetic anatomy look like? This is rest and digest. So our end result is to slow down the heart, right? But the anatomy is different. The first neuron is actually really long. And the second neuron is very short. Okay, and we're going to the heart. But now this pathway is going to slow the heart down. The neurotransmitter here is acetylcholine. And the neurotransmitter here is also acetylcholine. Okay, and here is our ganglion. And here's our ganglion here. All right, now I'm going to put this into a, excuse me, into a table form. Okay, but I had to draw it out so it made sense. Let's go back and fill out our table. Actually, let me draw my little things that I just did. Okay, so here's my anatomy. And now I'll do the table. Okay, so when we talk about function of the sympathetic, it's fight or flight, and parasympathetic, rest and digest. Length of the preganglionic axon. So preganglionic, that's before the ganglion. So that's your first flight if you got in an airplane. So in the sympathetic division, the first flight is very short, right? So think of you get on a plane in Santa Barbara. I live near Santa Barbara. This is Santa Barbara City College. So I go to that airport, and my first flight, I'm just going to LAX. That's a real short flight. But then my second flight is going to be really long. And let's say my destination is New York. All right. So this represents the sympathetic division, going to LA and then New York. Okay. Preganglionic short, postganglionic. In the sympathetic, the preganglionic, your first flight is really long. I think rest and digest is you get on the plane and you take a nap, you rest. Okay, so that's your first flight. You're gonna hook up your ganglion is maybe Chicago. I think it's O'Hare, right? Chicago, and or is it JFK? No, New York is JFK. Chicago's O'Hare. I don't fly enough, but and so then you get on your second flight at the ganglion, and the postganglionic flight is very short. Okay, and these are the different neurotransmitters released. All right, I hope that makes sense, that there's an anatomical part to the autonomic nervous system. All righty. Lastly, what is a stroke? Let's take a look at a clinical, something you might see in your work. A stroke sometimes is called a cerebrovascular accident. Cerebrovascular, so it's in the brain and it's vascular. You have a blood vessel that breaks, okay, and you have tissue death there. What are some causes? You're disrupting vascular supply and you get death of brain tissue. So you can get a stroke where it bleeds. Okay, so if a blood vessel is broken there, and then the blood, yeah, it's just kind of spilling out there. Or you could get a blockage of a vessel to brain tissue that would also result in death of brain tissue. Okay, I hope that was helpful. And it was a long lecture, so thanks for hanging in there. And hope it was interesting, though. And you're learning things about your body. All right, thanks for joining me.